to Science Night. Please stand by.
Welcome back to another edition of the Science Night podcast. My name is James, and with me, as always, is Steffi
Hey.
and Jason.
Hello.
We also have a special guest, an MD PhD student and host of your new favorite SciComm pod in plain English. It's Jamie Moffa. Thank you so much for joining us this night. Wait, hold on. The kayfabe is this is night.
I was gonna say it's really funny that this is the Science Night podcast, and I literally went from my bed to my chair, to record it.
People might listen at night. You never know. But
yeah, maybe.
Thank you so much for joining us. So
yeah, thank you.
Tonight we got some munchies on the mind, and in the second half, we're going to talk about how some tiny goggles might make mice happy or terrified. We have so much to cover, so let's get into it. Well, before we get into this science communication aspect of our science communication podcast, I think we got to talk to our new best friend, Jamie, who is, as we said, a student of, gosh, I was going to do something dumb, but I'm just going to say MD PhD.
Yes.
Who is an MD PhD student studying neuroscience, my favorite of the anatomical sciences. Of course, I have a long track record with definitely saying I'm not terrified by neuroscience. But anyway, Jamie, let's talk about you. Talk about what you do in neuro and your podcast in plain English, because I love this concept.
Yeah. So I am a neuroscience MD PhD student, which means I will be in school for the rest of forever. My family is like, when are you going to be done? I'm like, I don't know. But yeah, I am actually in the anesthesiology department at my university at WashU, and I am studying the neuroscience of pain. So how do our brains like interpret pain signals? How do our brains modify pain signals? Because your brain can actually kind of modify the pain signals that are coming up from your body to either make it, more painful, like, oh, you really need to pay attention to this, or less painful, like, oh, you've got this covered, or you're running from a bear and you don't have time to worry about that cut on your leg. So that's more or less what I study. And I do that by actually using gene editing technology. So if you've ever heard of CRISPR, it's a fun, relatively new way to edit genes. And I'm using that to delete a little protein in neurons in a part of the brain that responds to opioids. So you probably heard of opioids. You get them, when you go have a procedure done, or some people get them for fun. And that's a big problem, but that's not really what I study. And your body also makes them. Your brain makes opioids, and this is part of how it can control the pain signals that you're feeling. And so I'm going to use fancy CRISPR technology to delete the opioid receptor and then see what that does to how these mice are experiencing pain.
Speaking of pain, you're trying to take the pain out of neuroscience by taking all that jargon away with it in your podcast. So why don't you talk a little bit about in plain English?
Yeah. So I also have a science communication podcast. I have been doing that for 2.5 years now, which is kind of crazy. But I started in 2021, and the general idea of in plain English is, yes, exactly, to take sort of the jargon and the, all those like barriers to entry for the average person away from science. And it's not just neuroscience. I bring on experts in all sorts of different fields. We've had anthropologists, we've had like a mechanical engineer who's doing like literally rocket science. We've had on, so many different people in so many different fields, and they explain a paper in their field to two guests who know nothing about the subject. So they have to really try to make it approachable for these guests who are just, interested in the topic, but don't know all of the inside jargon. So it ends up being a really fun conversation, and really, we can get into some really interesting and like deep conversations about the science without it being so unapproachable, which I think is really cool. And I think is, something that was missing from a lot of at least science communication media that I'd seen, is that either it was this really like technical jargon and also usually paywalled scientific articles, or it was articles that you'd see in like pop science websites or like in newspapers that would really dumb down the science so much that it wasn't engaging with the material in a way that I thought was like very meaningful, or that would really bring people into the conversation. So that's what I wanted to do. I wanted to bring people into the conversation and tell them like, hey, you can understand this too. You can talk with the scientists about this too. You have enough knowledge. Us, the scientists, just need to like eliminate some of the barriers to entry.
I love that so much. Jason, I could see you swelling with pride using all the talking points from any communication training that you've ever done in your career.
Yeah, and I haven't even trained, Jamie. It's fantastic. I love the fact
pre-trained.
That's right. I love it. I love the fact that that this kind of information is getting out there as to how there's evidence-based ways to be more effective at communicating science or complicated scientific information to a general audience. And I love the fact that people are using those approaches, right? And so, yes, swelling with pride is right, although it's not pride for anything personal here. It's just for science with a capital S. I think it's fantastic.
I love it too, because oftentimes you see that the articles that make it into mainstream press, they kind of blow the context out of proportion to get it there. And then that's really hard to kind of get people, because then everything's conveyed as a breakthrough.
Yeah. And so people are expecting that science is advancing in these kind of unrealistic leaps and bounds. And then when they are faced with the reality of how science works, which is more gradual, more like, building off of steps that people have done previously, it kind of makes it seem like, oh, that's not as exciting, or, on the even more harmful end of that spectrum, like that isn't something that I should trust because it's not, what science has been shown to me in the media. And so, I mean, actually, the most recent episode of the podcast was not the kind of episode I was just describing, but I was interviewing another SciComm person, Will Nyam of the Reproducibility podcast. And we were talking about exactly that, because we were talking about how that kind of sensationalism harms reproducibility in science, because then you're like, scientists are also kind of internalizing this expectation to make these huge, giant breakthroughs.
Yeah. And then they aren't submitting funding for reproducibility.
Right, exactly.
Yeah.
Well, part of the problem there is that journals don't want to publish those papers, right? I mean, the publishing landscape, and I'm going to put on my journal editor hat for a minute, is just really different than it was even 10 years ago, right? And that's because, we used to rely on subscriptions to generate the revenue for journals, and now that's going away, and it's all coming down to these article processing charges that are being paid, right? And that is being exacerbated, not in a bad way necessarily, from an open science perspective, by things like the White House Office of Science and Technology Policies memo about sharing data that was recently written, about a year ago now. And the expectation that NIH is going to have to change the way that they're paying for open access fees for publication and those sorts of things. And so, it's just a different world than we were in. And I'm not really sure that it's fair to say that journals, or that people aren't submitting funding to do reproducibility or replicability studies. It's that the journals know that that's not going to bring in the APCs, right? These are article processing charges. And so they're not publishing those kinds of papers with the frequency that they should be, that we should be. And it's because they're not novel. They're not high impact, right? When in reality, like a confirmational study right, that shows reproducibility is a hugely important and impactful piece of information, right? And yet it's not going to get the same kinds of um, citation metrics that an original study will. And so journals recognize that, right? And so now we've got these tiers of journals that are becoming more stratified than they ever have been. And it's just an ugly side of science. Anyway, sorry, I'm gonna take my editor hat off now because that was totally bringing me down.
Take it off and lock it up.
Yeah, I should. Anybody here watch What We Do in the Shadows?
Yeah.
Sure.
Yeah. No, it's like Laszlo's
Okay, I didn't know
hat
where you were
made
going
out of-
quite with that, but
hat made out of witch's skin. Right?
Oh no.
Yeah.
I thought you were
so
just
take it
making a
off.
hard turn from
No,
talking
no.
about journals.
No, but I mean, yeah, there's certainly something about vampires in those journals as well, I'm sure.
Yeah.
True.
Amazing. You're doing an MD and PhD. That
Mhm.
is so much work, and I have so much respect for you. Because your expertise is so unique that you bring to like the research then. So why did you choose this path?
It's kind of an interesting story because I didn't start out as an MD PhD. I started out as an MD student, and I did my first 2 years of med school, and I was staring down the barrel of third year, which for people who don't know, that's when you do all your clinical rotations, and it's very intense. And I was like, I don't really want to do that right now. So I took like WashU lets you do this thing called an MD5 where you can just slot a research year somewhere into your medical curriculum. And so I did a research year, not in the lab that I ended up doing my thesis in. And my mentor for that research year was like, you should apply for the MD PhD. I think that you would really enjoy it. So I was like, sure, I will postpone doing clinical medicine for several more years. And also while I was in that lab, I kind of remembered like, oh, I really do like research. And I had thought that, when I was initially applying for med school, my research experience from undergrad was not enough, which wasn't true. I was just getting a touch of the old imposter syndrome.
Oh.
But yeah, so I remembered how much I loved research, and I applied to go into the MD PhD, and they let me in, and then COVID happened.
Oh, wow.
So my PhD is going to be a little bit longer than most because the first part of it was when nobody could be in labs anywhere.
Yeah.
And then at the end of your PhD, you slot back into your clinical years. So
Yes, yeah. So that's actually in a little over a year that'll be happening for me. So I'm currently doing experiments to like wrap up my thesis research, and then I'll be going back to clinic, which I actually now feel a lot more prepared for. So anyone who's younger than me is listening to this and you're like, man, I feel like my path is all twisty and turny. That's fine.
yeah,
You will end up exactly where you need to be.
That you couldn't have said that better. You're absolutely correct.
Amazing.
I've taken weird paths in my career as well, and I feel like I'm where I'm supposed to be. It's not where I thought I would be necessarily, but it's certainly where I feel like I should be. And I think that's really good advice. So at the end of the day, you will have spent quite some time in St. Louis.
Yes. 10 years.
10 years.
At minimum, unless I do residency here too, in which case much more time.
Right. So then I have to ask you the question because I'm married into a St. Louis family. And so I've been a regular visitor to that city for almost 30 years now. Emo's Pizza.
No.
No, okay, that's my reaction too.
Well, so originally
no.
I'm from the East Coast. I'm from Connecticut. And so Connecticut has New Haven pizza. We're really close to New York, so we can have like New York City style pizza. I've also come to love like Chicago deep dish pizza for when you really just want like an entire calzone, but as a pizza. But no, Emo's Pizza and the whole genre of cracker crust pizzas is bad. And also the Provel cheese is bad because it's not provolone. It's weird.
No, I agree with you.
What
So
is
it's
that?
interesting. So I'll come back to that. I will explain that in a second because it's nasty.
I'm gonna
But
make a lot of native St. Louisans mad with
that's
me.
okay. Me too. Welcome. You could just get right behind me. It's a long line. I've been doing this for a long time. I've been railing on Emo's Pizza for almost 30 years. For me, it's not the cracker crust that's the problem.
Mm,
It's the Provel cheese. Like the cracker crust, pizza shouldn't be a cracker. I agree with you. But
yeah.
thin style Chicago pizza, fantastic. So,
Yeah.
and it's got a pretty crispy, thin
Mhm.
crust. It's really the Provel cheese. So the Provel cheese it's a processed cheese, Steffi, that is a mixture of Swiss cheese and provolone, and it has the consistency of melted plastic, and it
Nope.
sticks to the back of your teeth, and it
Oh,
has this weird tang to it.
I don't like
It
it.
is straight up nasty. But I live in a house with two native born St. Louisans, right? My wife and my youngest kid are both from St. Louis, both born there. My oldest and I are not. We don't really care for Emo's Pizza, but like the two that were born there cannot get enough of it. And every time I go to St. Louis, every stinking time I go to St. Louis, we roll into grandma's house and there's an Emo's Pizza waiting for us for dinner. I don't have a choice. I don't have a choice. And I end up here's how much I dislike Emo's Pizza. I end up eating a salad from a pizza place
Oh
Oh
because
my
it's
God.
because it's only got a little bit of Provel on it. And
They
it's not-
put Provel on the salad too.
They do, but it's not melted, so you can remove it, right?
Yeah.
Anyway, all of this is to say, Provel is some kind of nasty byproduct of, I'm guessing, fission,
Yeah.
because
Oh!
fusion is clean,
Yeah.
right?
Oh no, this is
But
greasy.
fission is
Gross.
nasty, and it's got to be buried underground.
So you're
I
saying that
teach
not even a
both of those-
plasma could properly
No, I
melt a
teach
Provel
both of those
cheese?
classes.
I'm not
You
sure.
can't knock fission too.
I'm not knocking fission. I'm just knocking Emo's Pizza as a result of fission.
Oh,
Or some deal with the devil to make
Mhm.
the worst kind of cheese imaginable.
Are we sure that Emo's Pizza is not like it's not like a bit? Like people from St. Louis don't actually like it either, but now it's like such a part of their culture. It's like in Philly, we all pretend we like those like little corrugated pretzels that are super dry and bland, but like it's a Philly thing, so we got to pretend we like it.
Mm.
I don't
Right.
know.
Or
It
tasty
seems to be the
cakes.
case. I mean, tasty cakes are good, and I will fight you on that one.
I
Fair.
don't know. The people who I've met who are from St. Louis, they are crazy about it. And like it would be it would be a very large conspiracy for literally everybody who grew up here to all be pretending to like it.
Ooh, but now
That's-
let's
that
pretend it is a
said-
conspiracy to get those clicks up.
That
Yes.
said, it is also a very St. Louis thing to be in a cult. And so,
oh,
right? I'm sorry. I don't mean a cult. I mean a clique. I mean a clique. It's not quite cult status, although it's not too far behind that, right? Being in a cult is like being a Cardinals fan.
Ah,
Being in a clique is like being an Emos fan.
Or a Grateful Dead fan.
I
All
feel like
right.
Emos is
That
more of
you have overstepped your bounds here, sir.
my sister will have words with you. When she
right.
listens to the
Oh.
podcast, she will have words with you.
I'm
right.
gonna have to add some dead tracks to this
That's right.
as a mea culpa.
I've got a playlist for you.
Thank you. We just have to make this song. We have to make this episode 4 hours long so we can add one Grateful Dead song live.
That's right. That's right. All right. That was my question for you, Jamie. It was really about Emo's Pizza.
It
You
has to be
answered it.
about Emo's
You
Pizza.
answered it in one word, and that was beautiful.
Yes.
And it was
It wasn't
before I
even a
could-
word, it was an expression.
Right. And it
Yeah.
was before I even finished asking the question. Right. I just said Emo's Pizza, and I was going to ask a question, and you just said no.
So in my brain, I was like, please don't ask me where I went to high school because I didn't grow up here.
No. I understand. Little shout out to the Parkway North Vikings.
Jamie has officially passed the vibe check.
Woo.
Listen to their podcast in plain English. Oh my gosh. How did I step on English as a native English speaker?
I do this all the time.
Oh, sorry. The Philly accent popped out for a second. I said soft pretzels and I just started saying water in my head.
What are
Well, listen to their podcast in plain English. Well, we'll be back in just a minute with more of the Science Night podcast, but first a message from another show that I think you're going to enjoy.
Strange by Nature, your guide to the strange, weird, unbelievable, and improbable wonders of the natural world.
They make the sound by vigorously rubbing their penis on their abdomen.
Oh, yeah. Oh, my gosh. Then at night, they come out and crawl around your face and mate with each other.
Oh.
But
Sorry,
get this.
I would like to sleep tonight. As naturalists, let's face it, we find something dead, we go and we poke it with a stick. I did that with the
That's
deer
what-
like 3 weeks ago.
As you do. Rachel, no. You weren't paying attention. Blood and mucus.
Oh, right, sorry.
All right, this episode is going off the rails.
This is the quality
absolutely.
content people come here for. Strange by Nature podcast was chosen as one of the best science podcasts of 2021. Come join the fun wherever you find your podcasts.
So before we get into our first story, I just have a small trigger warning to any of our listeners that may enjoy cannabis. We will be talking about snacks. That being covered, it's no secret that cannabis increases appetite, and it's why it's often prescribed to cancer patients and patients suffering from eating disorders. But we didn't exactly know why it does this. But a team from Washington State University is attempting to find the answer. And when they looked at the brains of mice that were exposed to cannabis vapor, they found that a group of cells near the middle of the brain called the arcuate nucleus are turned on, and there was also an effect to the hypothalamus, all of which are involved in appetite regulation. So basically, cannabis can turn off the signal that is turning off your appetite. Now we're going to talk to a person who actually knows about neuro. Could be like, James, you've read that totally wrong, and this is why you're neurophobic. So, what do we think about what do we think about these munchies on our mind?
That seemed like a good description to me. I thought this article was super interesting. And I've never really known what causes the munchies. It seems like scientists didn't either until recently. But yeah, what I want to know is where these neurons are in my brain, because I've actually never had this happen to me.
Right. So I guess that's the other thing we have to say. This was a controlled study in mice.
Right.
I guess the controlled study in humans is more of a self-selected thing.
Right.
But you're right. You know, it's we're looking at a very controlled vapor in a very controlled situation with very controlled electrodes inserted into mouse brains.
Right.
So I guess we have to throw those caveats up.
Firstly, you can't go around inserting electrodes into people's brains. You'd kind of have a problem on your hands if you tried.
Not legally, you can.
Actually,
No.
not even ethically. You can put electrodes into people's brains. You just have to have the IRB approval to do it.
That's true.
I was next door to a lab that did that regularly when I was a postdoc. So,
Well, people who didn't already have so I know people who have like Parkinson's, for example, can sometimes have implanted electrodes. And so then you might do studies on them because you already have the electrodes. So you might as well use them for something other than the Parkinson's. But you can just put electrodes into people's brains, like who don't already have them.
You can, not as long as they're not permanent indwelling electrodes, right?
Okay.
There are things that can be done. But it has to be justified, right? And it has to go through institutional review board approval process, which always includes someone from the general public as well, because if we're doing things that manipulate people, they have to be ethical.
Mhm.
And having someone who is outside of the field looking from that 30,000-foot view is always helpful in understanding what the ethics of the public are.
Mhm.
Because scientists' ethics don't necessarily always align with public ethics. That's true. Public ethics. When I say that, there's no one public ethic either, right? I mean, parts
Please
of the
see, for
public.
example, politics.
Right.
I was gonna
Exactly.
say,
Exactly.
so much.
Right. Or anything, right? I mean, let's see, for example, cannabis, right? I mean, your state, Missouri, is legal.
Yeah.
My state, Indiana, not so much, but every state around me has the munchies. So
We're coming for you.
that's right. And I would say probably a large proportion of Hoosiers also have the munchies because they live near the borders of those states.
Right. Yeah. Pre it being legal in Missouri, we just go over to Illinois.
That's right. That's right. So I am actually just struck by the fact that you said you've never gotten the munchies.
So this is a casual interest of mine. I don't know how I'd ever go about answering this question, but my brain does not respond the same way to cannabis that my, friends who, will smoke occasionally do. It sucks. I just skip the like chill phase entirely and go directly to anxious and paranoid. There is no like fun stage for me, and so there's also no like I'm relaxed and eating food stage.
Yeah, that makes sense. You're in fight or flight mode.
Right. So like why? What's going on up there? I don't know. Maybe eventually someone can take my brain apart and be like, oh look, you are missing a whole kind of like receptor for cannabis that means that it just sucks for you.
All right. You opened this door, so I'm gonna jump through it. And as a gross anatomist by training, I'm often, and I think James probably deals with this more than I do as a trained mortuary scientist, but I'm constantly thinking about, would I be comfortable donating my body for dissection? Knowing what happens in the lab. As a neuroscientist, would you consider donating your brain posthumously?
Yes. This is like not a very difficult question for me. Also, I have also done gross dissection, being also an MD student. So
right, sure,
we have to do dissections, and we had my like anatomy group and I had made jokes about like planning to get tattoos that would show you like where to make an incision, or like, oh, the brachial plexus is under here, to make it easier on the future medical students of the world.
So will you donate your body to a gifted body program? Is that the plan?
Possibly. I hopefully am a long way off.
Of course. I don't mean to rock the boat by any stretch here. I'm not trying to tip the balance in a particular direction. I just wanted to know sort of ethically, what are your thoughts on that, right?
Yeah,
so I don't know. I don't know how I feel. I'm just going to throw that out there.
Yeah. Like the other thing is like on my driver's license, I am a like donor. So I guess if something were to happen in the way of like a car accident or something, that would probably be what happens with my organs instead. But I hope also, if in the very, sad future that happens, people in my lab and surrounding labs who are not me, but I hope to do this someday, actually study human brain tissue or human nervous system tissue. And they get it from organ donors who, after the whole procedure has been done to get all of those vital organs that other people badly need out, they go in, they take out like sensory neurons that sit right next to the spinal cord to record from, and bring them back to the lab and record from them, and can actually learn about how human neurons work, how human sensory neurons work, all the different like really nitty-gritty properties of these neurons that can help us understand like the ways that they transmit the signals from the periphery up into your brain. So that's super cool. And if that were to happen and most of my other organs go to people who need them, I hope that my nervous tissue goes to people who can record from it and be like, oh, that's fun.
Yeah, I get what you're saying.
I have a question. What does record from them mean?
Oh, whoa. Okay, so this is actually one of the craziest things, and one of my favorite things about neuroscience is if you keep brain and like nervous system tissue in the right kinds of liquids to keep getting them oxygen and nutrients, you can keep the cells alive for several hours after they exit the body of whatever organism that they came from. And so then you can take a little like electrode, and under a microscope you can, and using like a little manipulator thing, you're not doing this by hand, you can take an electrode and you can move it down until it's on, you can visualize the cell through the microscope, and you can move the electrode down onto the cell, or you can put it in, like break into the cell, and then you can actually, neurons, their language is electricity. They use different charged atoms in order to change basically, the electricity that's moving through them. That's kind of how they send information, is through these like electrical potentials. And so you can actually record those through this electrode, because it's also conducting electricity. And you can find out, like, if you put more charge into the neuron, like how excitable is it, how long does it take, how much of this charge does it take to get the neuron to like send a signal. Or you can find out like what's the general resistance of this neuron, how basically resistant is it to getting charge put into it. And you can learn all these different kinds of properties, which can help us then understand how these neurons are working to send signals when they actually are in an organism. So that's super cool, is you can literally just plop an electrode down onto a neuron and then find out its inner life.
That's amazing.
Yeah.
So you have to supply the charge or the potential to it? It's not stored?
So this is can I explain electrophysiology in plain English? Let's find out. So kind of, yes. If you think of, you can think of a neuron as like part of, if you're more electrically minded, you can think of it as part of like a circuit, like the kind of circuit that, lights your light bulb, lights in your house or whatever. And so you can either kind of monitor what's going on in the neuron at baseline and just see, like, you're just trying to keep it at a neutral state. So you're not really doing much of anything to it. You're not putting any charge into it. You're not doing anything like that. And you're just seeing what it's doing. And it will do things at baseline as long as it's alive, which you're keeping it alive through all these solutions. Or you can try to like manipulate it. You can try to change what its baseline is artificially by injecting this charge, like making ions move, and then see how the neuron responds to this artificial change in its baseline. So hopefully that makes sense.
That's amazing.
It's great.
Wow.
This is the part of neuro that fascinates me and intrigues me and makes me want to get better at it. It's just so mouse forward. It's so mouse and rat forward that I can't. I really can't.
Yeah. Well, and I primarily work with mice. It's the easiest way to do a lot of the things that you want to do in neuro, because, first of all, mice are small, they reproduce quickly, and people are somewhat less concerned. Like, you still have to go through the IRB, and there's still ethics, and you shouldn't like mistreat your mice, and that's a very big problem. But also you can do a lot more things to them than you could do to people. And then also something that's relevant for my work is that people have actually made, like, they're not mutant mice. There's a better way to put this, but they've messed with the genomes of mice. They call them transgenic mice. And so you can delete genes, you can add genes, you can change what this gene, you can change the sequence of this gene. You can do all sorts of weird genetic stuff to mice, and it's actually really helpful in being able to more specifically get at like what's going on in this specific gene in this mouse, and can we like mess around with it. So that's a reason for the mice, but I agree, and a long-term goal of mine is to actually do some of this work in people.
There's a faculty member with my same name in that field. I don't know what field, medical field somewhere. And so I would always get her email for custom knockout mice and
Yeah.
like holiday deals.
I
Yeah.
love that.
Yeah.
Hold on. I think
Yeah.
we gotta go back to Steffi. You can't just say there's another Steffi Deem who is also a scientist.
I think they go by
out
this
there?
Stephanie. Yeah, Stephanie
Oh,
Dean. I know.
okay.
I've never
See,
met them.
that's why they're not in Fusion. That makes
Right.
sense now.
Yeah.
Yeah.
they're-
Because you're
Yep.
not gonna be Stephanie and be a fusion scientist. That's
Mm-mm.
Oh,
I don't even know what that means, but I think I agree. I don't know why, but I do.
yeah, exactly. You don't know why, but it is true.
It's true.
Yeah.
Stephanie is a biologist. Steffi.
Yeah, that's
Yeah.
true.
Exactly.
I think we can all get
Changes
behind that.
everything.
That's true. More specifically, Stephanie's probably a field biologist, right?
No,
No.
they're rolling in mice.
If you're ordering knockout mice, probably not.
Well, right, except that the name Stephanie. I think I can probably name 6 or 7 Stephanies I know who are field biologists, which is just
Ah, okay.
random.
Mm. So this is the outlier, Stephanie. That's more of an experimental biologist.
Mhm.
Mm.
Yeah.
I mean, there has to be an outlier, right? Makes sense.
Right.
You know, in my head canon, this Stephanie was a field biologist. Probably in the American Southwest. I'm gonna say looking at what do they call them? Fence row lizards, I think. And just they found out that the dry arid climate wasn't for them.
Mm.
But they really loved the bench research aspect when they got into it, and now they just they have a long career using knockout mice. Probably the Brownsville rat, I think, would be a good mix for them. Looking at urine analysis and that sort of thing.
This is
Yeah.
detailed.
So
Yeah.
really quickly before we move on from this, because I agree with that story, I feel like that's the story that careers are made on, right? We have introduced a new piece of jargon inadvertently into this. When we were talking about transgenic mice a little while ago, Jamie introduced that concept.
Yes.
Mice that their genomes can be changed. And now we've started talking about knockout
Oh,
mice.
sure.
And we didn't explain what that means because there's also knock-in mice, right?
I don't know what it means.
so
and
knockout
I got so
mice.
many ads.
Yeah. That's precisely why you got the ads. I mean, I get all those ads too. I don't know what they mean. But when you knock out a gene, you remove the function of that gene from, whatever environment you're trying to remove it from, whether it's the whole genome, whether it's a particular cell type, whether it's, whatever it may be. And you can also knock in genes, meaning you can turn on genes that are not there or put genes in that were not there, or turn ones on that are not active in that particular area. And so it's really interesting how you can manipulate. And you do a lot of this actually with CRISPR, which is
Yay.
what Jamie spoke about earlier, right? So it's amazing to me how much this field has changed since I've been a faculty member for 16 years, right? Because when I first started, transgenic mice were really expensive to make. And now they are not that expensive to make anymore. I mean, they're still expensive compared to like going to the gas station and, buying a pack of gum.
Right.
But
if you want to make a new transgenic mouse, it's still annoying. Like if
Correct.
there's not an existing mouse that does what you want it to do and you have to make a new one, it's like a whole PhD project and it's also
Right.
a bad idea.
it's a terrible idea for a PhD project for sure.
Yes.
Because at the end of the day, you've learned how to do one thing,
Yep.
right? Really
And you've
well.
had to do it over and over and over again.
That's right. That's right. It doesn't really set you up to succeed unless your job is going to be to make new transgenic mice for the rest of your career, which
It
you
could
can
be.
make. Yeah, you can make a career doing that. But it's not usually what people go into bench science to do,
No.
right? They have, pie in the sky dreams. So not rodent under the microscope dreams.
Yeah.
Another
So-
thing you said actually that really intrigued me, Jamie, was you're talking about why you use mice instead of humans, right? And one of the things you said is that you can do a lot more things to mice than you can do to humans. And I want to push back just a little bit on that.
Mm-mm.
You're not wrong, but you're also not entirely correct in this one. And that's because you can actually do the same things to animals that you could, or to people that you can do to animals. It's just harder to get them approved to do in animals because animals can't consent, right? And so you can convince a human to let you do anything. That's an ethical issue in and of itself, right? Coercion cannot be part of this. But they can consent. A mouse cannot consent. And so to be able to do the kinds of things in an animal that you would want to do in a human or vice versa, it's always harder to get it approved to do in an animal because they can't consent for themselves. But you can still do very invasive things to humans because they can consent. It just has to be a very good reason for it.
Yeah.
So this goes back to that discussion about electrodes in the brain, right? And whether you can implant electrodes in the brain that are permanent or not permanent. And you brought up this idea of Parkinson's patients, right? Who have deep brain stimulation. So they have these electrodes embedded deeply into their brains, and they have these two like knobs basically on the top of their skull where you can see the healing has happened where these two electrodes have been placed. To be able to do that required the research approvals to be able to do that the first time, right? That is incredibly invasive. And you don't get any more invasive than something like that, sticking an electrode permanently into someone's brain. But the benefit of that deep brain stimulation, at least from the experience that I have had, which was I had a professor in grad school who had this procedure done, and it basically turned back the symptoms of Parkinson's by about 10 years for at least a few years. And quality of life, right, improved incredibly. And so that's why you can do really invasive things to people, but you have to get them to consent to do it first, right? So it's not that you can't do those things in humans, it's just that it's harder to get them to consent, right?
Right. And obviously there are also drawbacks to using mice.
Absolutely.
The like bigwig researcher in my department loves to say that mice are not tiny humans. You can't. So that's relevant actually to the article. Which I will help you all by bringing it back into the article. Is that mice aren't tiny humans, and so like you can learn a lot of really interesting things in neuroscience and in biology in general by looking at mice, because they are mammals, so there are some similarities in terms of the structure of their brain and nervous system. For example, they have by and large very similar brain structures to us. They have the same kind of brain spinal cord layout, but there are differences. And so one of them that I was kind of gonna bring up that is kind of crossing between like neuroscience and like psychology is that like the setting of like these mice getting the cannabis is very different from when most humans will like consume cannabis, and why most humans will consume cannabis. And so like you can't really replicate people hanging out at somebody's house or at a party and smoking in mice, right? You're just kind of vaporizing the cannabis at them. And it kind of does the same thing, so it can maybe tell us about like part of the reason why that people get hungry when they consume cannabis, but it also can't tell us the entire reason because they're mice.
Ooh, I get to bring anthro into it now.
Yay!
So we're learning the circuitry of how cannabis affects the literal mechanisms in the brain. But when you kind of bring that out to humans and you add in the culture of the consumption and all the practices around that, whether it be recreational or in certain cultures part of some kind of community experience or religious thing or something, there's all these other factors to it. So you're totally right, where like we know that the cannabis is going to affect this clump of cells that kind of works in this mosaic, but then there's all this other human stuff that we put on top of it too, which was what I was thinking about when I was reading this article, because I was just trying to not think about mice, because boy, am I afraid of those little guys.
Oh no.
What I was thinking about actually in this when I read this story was when I was taking we had Carol Ward on the podcast not that long ago, and we were at Gen Con, right? And she, if you recall, she is
Y'all were at Gen Con?
Yeah.
Oh, yeah, we go to
every
Gen Con every
year.
year.
Were you at
Do
Gen
you
Con
go to
too?
Gen
I
Con?
was, yeah.
Missed
right.
opportunities. Did you go to the Science of Science Fiction presented by Indiana Sciences?
Oh man, I should have. I don't remember what I was doing at the time, but I
We
saw
do
that it
a
was
panel.
We did the
Yeah, that's
panel.
us.
We
Yeah.
were there. You could have got
Yeah.
a koozie.
Oh no.
Are you going this year?
Potentially. It's
Okay.
Gen Con is falling like right after a big conference that I'm going to in Amsterdam and right before my brother's wedding. So
Ah,
Oh, so that's a
Ooh.
perfect mini vacation between the two.
I can just
What
go
I'm hearing.
directly from Amsterdam where I'm presenting
Yeah.
a poster, immediately fly back to Indiana so that I can go to Gen Con and then
Yeah.
fly to Connecticut so I can see my brother get married.
It's like on the
Perfect.
way.
Yeah,
Yes.
it sounds perfect. It's totally
Yeah.
on the way.
And
Totally on
definitely
the way.
not in the middle of the country.
Right.
Sure.
It's so good.
Okay, anyway, you were at
Anyway,
Gen Con.
we had Carol Ward on the podcast. Actually, I can't even remember where I was going with that now. Now that I'm so excited that we're going to potentially see you at Gen Con. What were we talking about? What were we talking about?
You were
I remember. So I
anthropology.
remember. So this reminds me of this. When I was reading this story, it reminded me of when I took Carol Ward's Intro to Human Origins course when I was in an undergrad. And I was studying really, really hard for this final exam. And my roommate, just want to give a shout out to Buff. That's what he calls himself. What's up, Buff Daddy? My roommate in college was consuming cannabis at a rate that was, probably not safe for like an elephant. And but I was studying really hard for this final, right? And I went in and that was my area. I know that stuff. Or I used to know that stuff like the back of my hand, and I bombed the final, right? And it's because of all of the secondhand smoke that I was exposed to while I was trying to study, and then didn't take my exam in the same state, right?
Ah.
I think there's some truth to that. And so this, vaporizing the smoke for these mice reminded me of secondhand smoke. It isn't, so there was a real point to this, and that is that it wasn't the active consumption of cannabis in this case, but the passive consumption of cannabis that was actually happening. And so I'm wondering if I wonder whether that makes a difference, right? At least in humans, right? If you are intentionally doing something versus unintentionally doing something, is there something that's going to modulate a response, right? Deeply rooted in our nervous system somewhere. And I suspect the answer might be yes, right? I mean, we have all of these things, as you alluded to earlier, Jamie. We have ways of tamping down our perception of pain and tamping up our perception of pain, depending on a situation and whether it's worth our effort to know it's there or not know it's there in that instant. And I wonder if there's something to that too with appetite, right? Like you have to come to the situation ready to be to have your appetite stimulated or not in order for it to work.
So this is something that I was thinking about when I was reading this, which was that they kept bringing up the use case of treatment for eating disorders. And I would need to do more research on this, but I'm skeptical for this reason. And also for the reason of you would have to get the patients to consent to consume something that they think might make them eat, which would be a challenge in and of itself. But I am skeptical that if they are not coming to the experience with the frame of mind of like wanting to have the whole experience of having the munchies and everything, that would actually work out well. And I remember talking to, and so this is why I need to look into this more, because I'm not entirely sure how common practice this is. Because I remember asking somebody if this was like an eating disorder treatment, like physician, if this was something that they did. And they're like, sometimes, but not always, and mostly for reasons of consent, right? Like you're telling this person who like does not want to eat that you're going to give them something that's going to make them want to eat. Like that's going to be really tricky unless they're at a particular place in their like recovery and their mindset. But yeah, I'm not so sure, again, because of that whole the passive versus active consumption and the like mindset of it, that would be as effective as these researchers seem to think it is. And so to me, it came across as like, oh, this is the way we can tie it back into something that people know about and understand, but maybe doesn't have that much grounding in clinical practice.
Probably also depends on how you frame that, right? Like,
Right.
hey, please take this THC supplement. It's going to help stimulate your appetite versus why don't you take this THC supplement? You're going to blaze through this plate of nachos, right? Like very different sounds, or very
Right.
different approach, right? And I'm wondering if there's something to the way that is conveyed as well, right? People are
Right.
complicated, right? And so you never know what's going to tip the balance in one direction or the other.
Right, exactly. And then, you could accidentally like trigger a pretty bad experience. Like if someone had given me THC as like a treatment and I was like, discovered in the worst way possible, that it just makes me super paranoid. I'm like, I'm never doing this again. This is awful.
I have really weird reactions to drugs too, like just in general, all of them. So yeah, I'd be right there with you.
All right, we need
so
to.
there's another story that we were going to talk about, but then Jason went on his pizza rant where they created a VR rig for mice and rats to experience the outdoors, but in an internal environment. And Jason, I'm sure you're going to riff on this cannabis story right now, right?
Yeah, well, I just wonder why there isn't the follow-up study where you vaporize cannabis and then give the mice these little tiny VR headsets and see if they get better at Overwatch.
Oh,
I don't understand why that study hasn't been done, right? I mean, we're talking about making little tiny VR headsets for mice. That's amazing. It's amazing because the researchers are actually trying to reduce the number of variables they can't control for in the wild, like in a field environment, right? But as someone who has done field work with electrophysiology involved, it's not impossible to do this, and I don't.
right.
I didn't understand why there was suddenly an inability to study the things in the wild that we'd like to study. So we have to build a virtual environment to simulate that. Was like the thrust of this paper, right? I mean, I think it's amazing that we can do the kinds of things that we can do. And I have no problem doing science for science sake. But this gets back at the question as, about sort of impact, right? There's a headline about this story. But at the end of the day, it's way too premature to say anything about the utility of this kind of work, right? There's probably a very good use for this work, but just to point out that you can build headsets, VR headsets, to simulate a natural environment for mice seems like that horse is out of the stall way too early, out of the barn way too early on this one. And this is the problem with sort of that sensationalism of science, right? I mean, people are going to see these kinds of studies, and it's, again, I am not knocking the science. I am sure there's a very important end goal for this work that just wasn't reflected in this particular story because we're not there yet, right? But the public then sees those things, right? And they get confused. And then when you get politicians saying, why are we funding this kind of work, right? And calling out individual grants by name on the floor of Congress, right? We get a lot of mixed messages that undermines the importance of science to evidence-based policy decisions. And so it's frustrating to me. I love that this has been done. I don't like that this is getting press yet because it's not there yet. It's not ready for prime time, right?
Right.
Not everything is a huge leap forward. This one is a huge leap forward for stoner mice, but that connection was missed.
One small step for
Right.
Yeah. Next, is the follow-up study going to be miniature tape recorders so that they can be ingrained into the culture of creating their Grateful Dead live tapes and start getting into that community? Like maybe a little backpack they can put on with a cassette recorder on the back.
We
Those
brought it
already
back to
exist,
Grateful
actually.
Dead.
They already
Yeah.
exist. Those things already exist. Little backpacks that they can wear, that can hold weights, that can hold
Okay.
telemetry equipment
There's-
so that
yeah.
you can, you know.
There's a
Sure.
lab on my floor. One of my friends works in it. That they give the mice backpacks and it holds. They do like IV self-administration stuff. So the backpack holds the like little IV tubing, and they can press a lever and get, I think it's opioids, just through IV. But yeah,
Right.
mouse backpacks. It felt so. Yeah, what this study felt like to me is it felt like someone. It's like inside baseball, right? It's like they took something that like researchers will use. And I actually know some people who use like similar mouse VR type stuff because it is time prohibitive in addition to like difficult to sort of figure out the logistics to bring a mouse into a naturalistic setting and have it do its thing, right? You would have to find somewhere like that and go bring the mouse there. But it felt like this is something that's inside baseball, that's like something that is important for researchers, but it sounded cool enough to a science editor at this website that they were like, let's tell everybody about it. Which, I support telling the public about science, but yeah, I think I agree with Jason that it's kind of devoid of the context that it's useful for, and it's just like, look at this cool mouse VR thing and not look at all these cool questions that we can answer because we now have this mouse VR thing.
Totally agree. And I was really sort of off put by the setup that we can't do things in the wild, right? Well, that's not entirely true. It's time intensive. It's energy intensive to do those things. And sometimes it is easier to do something like this than it is to find an environment that would be perfect to run the experiment. But trust in the process can be lost when these black and white sort of statements are made. That really frustrates me. There's a lot of nuance, and we talk about nuance all the time here. And that nuance is what makes the discussion of science with the public so much more enriching. But it's also what turns off the public immediately because they see contradictory things, day to day, right? Like coffee is great for you. Coffee is terrible for you. Coffee's not as bad for you today as it was yesterday, but it's still not great, right? What am I supposed to make out of that, right? Not everything, like you said, Jamie, is a headline here, right? Sometimes it's just incremental changes in the nuance. And when we don't have that context there, we miss out.
Maybe this is actually the internal arc of our character Stephanie Deem, bench scientist, where she was doing her mouse work outside in a park and she was sick of controlling for falcons. So she went indoors and created a VR rig for her knockout mice.
Many of the articles have an illustrator's whatever vision of what this looks like, of a mouse wearing VR glasses. You have to dig really hard to see what it actually is.
Yeah.
And this gets me all the time for these articles. There's like, and you'll see this for fusion. It's like artist's conception of a magnetic confinement device for fusion. I'm like, we have them. You can use a picture of the real science.
It's hard to get consent of the mouse to be photographed, though.
Actually,
this is actually true. Is that I can't take pictures of my mice. And
really?
Yeah, mostly because of like PETA reasons. That like if you start sharing photos of mice on your social media, then like all of the animal rights groups will get wind of it, and then all of a sudden you'll have massive protests outside of your university because you posted a picture of a mouse with like a little electrode sticking out of its skull, and people got upset about it. So you have to get specific permission to use these photos in, like, if you need them for a paper, for example. but you can't
Oh.
share them publicly.
Right, but you
Okay,
can
that's
get
good
an
to know.
illustrator to
Draw
draw
it.
what that looks
Yes.
like, and that is perfectly fine, and
Yep.
it avoids a lot of those problems, right?
Can we at least get a picture of the VR goggles? They don't have to be on a
So
mouse.
I did an internet search for it, and I found a picture of a mouse with the VR goggles, and every single time I found it was just Joe Biden with his aviator glasses on. I'm telling
No.
you.
God. Well, I think the fact that we're talking about Joe Biden and aviator glasses means we've come to the end of another edition of the Science Night podcast. But don't worry, we got more coming your way, unless we get canceled on the internet for this episode. But
For
anyway,
all reasons.
I think that's the reason you should be following us on social media. If you want to follow me, I'm on Twitter, I guess. I'm still calling it Twitter. I can't
Yes.
stop calling it Twitter. I'm on Twitter at James_Reed3, where you can see me switching gears to basketball because the Eagles let me down yet again. So anyway, Steffi, where can everybody follow you?
I don't know, that X Twitter thing, Steffi Deem, Instagram, Starshipin. I also started a new Instagram account that's more Fusion all the time.
Oh.
Fusion.deem. So there you go.
Jason, where can we find you?
You can find me on what used to be called Twitter. Is that better?
Yeah.
At Oregon JM. But enter at your own risk.
Jamie, tell us all about where we can find you and your podcast.
So I am on Twitter. I use my personal account more than I use my podcast account. So my personal is That Brass, ThatBrass. My podcast account is Plain English Sci, PlainEnglishSci. And you can find the podcast in plain English if you search In Plain English Science in any of the places you get podcasts. And we have a website where we post the papers that we discuss at inplainenglishpod.org.
Well, you can follow the show at SciNightPod and visit our home on the web at SciNight.com, where you have links to all of the articles we cover, the people we talk to, and of course, our merch. There's so much to see. You can see it all at SciNight.com. Jamie, thank you so much for coming on with us today. We had a lot of fun talking to you.
Thank you. This was a really awesome way to start my day or end it. Wait, hang on.
Yeah.
Yeah. It's a liminal space. Now, once you enter Cyanide, there is no night or evening or morning or afternoon. There just is the pod. And I think that means we're going to be back in 2 weeks with a new episode. So until then, have a great whatever time it currently is. It may be night, it may be morning. I don't know. Depends. Maybe you're eating lunch. Maybe you're driving in your car. And if you're driving in your car, just pay attention to the road, please. There's so many accidents when I get that I am hilarious, and I get that you're gonna get a laughing fit out of